Hello, my name is Shui Ma, and I am an assistant professor at Seattle Children's Research Institute. Today, I'd like to share with you a recently published collaboration with Sriram Chandrasekaran at the University of Michigan, Bree Aldridge at Tufts University, and David Sherman at the University of Washington, focused on predicting multi-drug interactions in Mycobacterium tuberculosis. This project is motivated by the urgent need to design multi-drug regimens that treat TB, an effort that's complicated by the astronomical number of possible drug combinations. As this figure shows, there are over 2 million ways to combine 50 compounds into a 5-drug regimen, rendering experimental prioritization of combinations intractable. We sought to develop a computational approach to efficiently and effectively prioritize drug combinations by accounting for the nonlinear interactions of synergy and antagonism between these drugs. In collaboration with Sriram Chandrasekaran and Suraj Jagpali, we developed a tool called INDIGO-MTB that uses machine learning to predict synergy and antagonism between multiple drugs, using patterns of gene expression measured from TB response transcriptomes during monotherapy in vitro. In collaboration with Bree Aldridge and Jonah Larkins-Ford, we found that INDIGO predictions correlated strongly with experimentally measured pairwise and three-way drug interactions. INDIGO is also able to qualitatively assess synergy and antagonism with high sensitivity and specificity. In addition, INDIGO interaction scores significantly correlated with 2-month sputum conversion rates, a metric of clinical regimen efficacy in humans. Regimens predicted to be more synergistic were also found to be more efficacious clinically, and regimens predicted to be more antagonistic were found to be less efficacious clinically, even though the current INDIGO model is based on in vitro data only. This suggests that INDIGO is tapping into important biological signal, which should be improvable with models tailored to in vivo infection contexts. Finally, INDIGO's predictive gene signatures provide information about the genes that mediate drug interaction phenotypes in TB. INDIGO predicted that the transcription factor RV1353C would shift TB drug interactions for several drug pairs towards synergy when induced. To test, we experimentally compared the drug interaction scores for 2 combinations that were predicted to shift. In both combinations, inducing this transcription factor shifted the interactions towards synergy, and disrupting the transcription factor shifted the interactions toward antagonism, all consistent with the modeling predictions. To test specificity of the modeling predictions, we further measured the interactions of a drug pair predicted not to be influenced by RV1353 expression, and found that genetic manipulation of RV1353C expression had no effect on the drug interaction outcomes. To summarize, our model accurately predicts interactions between multiple anti-TB drugs in vitro, and predictions also correlate with clinical efficacy in humans. In addition, INDIGO gene signatures can be used to identify regulators of drug synergy. Our next steps will be to refine INDIGO-MTB for predictions in vivo infection, and to adapt INDIGO to predict how drug toxicities interact in the host. To close, I'd like to thank the many collaborators who made this work possible. This project was completed while I was a postdoc in the David Sherman lab. I'd like to thank my mentor and former lab mates for their support and experimental help in this project. I'd also like to thank the support of our collaborators in the Chandrasekaran, Aldridge, Hernandez, Jacobs, Cole, and Stirling labs, as well as our funding. Thank you for your attention.